Before we start, I'd like to welcome everyone, but let you see here, this meeting is going to be recorded so that we can post it on the WCMM website later. So I'd like to welcome everyone to the Wallenberg Center for Molecular Medicine's Clinical Arena Lecture here at Linköping University. I'm Rosalind Simon, I'm a lecturer here at LiU, and I'll be hosting the arena today. In general, the central aim of the WCMM arena is to highlight collaborations that bridge researchers from the Faculty of Science, Engineering, Medicine and Health, and Linköping University Hospital. Today, we're honored to introduce two esteemed Wallenberg researchers who teamed up for an interesting project to shed light on pain signaling. I'll start by introducing Dr. Jackson. He received his PhD in 2003 from the University of Alabama, Birmingham, where he genetically engineered and studied mouse models of neurodegenerative disease. He continued in this line of research for his postdoc with Sue Lindquist at MIT, Massachusetts Institute of Technology, and then after that, following that, he started his own research group at the German Center for Neurodegenerative Diseases in Bonn in 2011. In 2018, he moved his lab here to LiU to become a fellow at the Wallenberg Center for Molecular Medicine in the Department of Biomedical and Clinical Sciences. In collaboration with Dr. Jackson, today we have Dr. Olausson, who received his medical degree at the University of Gothenburg, where he earned his PhD in physiology. And in 1998, he became a postdoctoral fellow at the Montreal Neurological Institute in the Department of Anesthesiology and Neuroscience at McGill University in Canada. Since then, he's held positions as professor in clinical neurophysiology at the Universities of Gothenburg and as a visiting professor in Western Sydney. And now he's here in Linköping University at the Department of Biomedical Clinical Sciences as well, at the Center for Social and Effective Neuroscience, CSAN, where he recently this year was appointed as a Wallenberg Clinical Scholar. So welcome to you both. Looking forward to hearing about your collaboration. I'll hand it over to you.
Okay. Should I start, Håkan?
I think so, Håkan.
Yeah.
I'll go on mute.
I share my screen then. Can you see this? Yeah. So I'm going to talk about the project that has been ongoing for, we started in January 2020, and it's a collaboration between clinical and basic sciences around a newly discovered pain system. And I work clinically as a clinical neurophysiologist, and almost every day we get referrals for investigating patients with pain problems. So this is a typical patient, 56-year-old woman. She has shoulder and neck pain since 5 years. She works as a teacher, but now she's on half-time sick leave because of her pain. And all clinical examination is almost always normal. We do all our standard clinical neurophysiology tests, but it's almost always normal. Can't find anything, and you can't really see from the outside that she's in severe pain. And standard pain treatments with non-opioids typically have minimal effect, and many people turn on to start with opioids eventually. And this is how she describes the situation. It's like a stabbing pain from the neck and shoulders, which she says is driving her crazy. And this is a huge problem. About 25% of Swedes suffer from chronic pain. And we think we have made a fundamental discovery about a year ago. And the textbook view is that, so I'll try to find a pointer here. Textbook view is that touch is transmitted in very fast peripheral nerve fibers, whereas pain is signaled in slower nerve fibers. Touch is signaled in large myelinated A beta fibers with a conduction velocity of around 50 meters per second, whereas pain is signaled in slower, thinly myelinated A delta or unmyelinated C fibers. The A delta fibers have a conduction velocity around 10 meters per second, and the C fibers 1 meter per second. But we have found that there are, in humans, pain fibers with as fast conduction velocity as the touch fibers. So we call this fibers ultra-fast pain fibers or ultra-fast nociceptors. And we discovered these ultra-fast nociceptors with a technique called microneurography. It was developed in Sweden, in Uppsala, about 40 years ago now, more, 60 years ago. And the way it works is that you insert a thin needle through the skin and into the nerve. And when the tip of the electrode is in contact with a single nerve cell, you can record action potentials from that nerve cell with a high signal to noise ratio. And the microneurography here in Linköping is performed by my colleague Saad Nagi, who is a researcher. And this is how it looks like. So you can see the action potentials here from a single nerve cell. It's highlighted here. And it's like eavesdropping on single nerve cells. And what we want to do now over the next 5 years is to characterize the system of ultra-fast nociceptors. We want to see where in the brain they project, where in the spinal cord they project, and we want to look at the peripheral transduction mechanism in the skin. And we think that this might lead to new knowledge, and textbooks has to be rewritten. We hope that we will find a way to improve diagnostics of chronic pain, and the ultimate goal is, of course, to improve treatment chronic pain patients. So I divided this into 3 projects. The first project is to look at touch-evoked pain. Second project is to see where in the spinal cord and in the brain the ultra-fast nociceptors project. And the third project is to try and find a way to measure from the pain system some kind of objective measurement of pain. So if we start with the first project, touch-evoked pain, I think many of you have experienced that. For instance, following a sunburn, light touch on the skin becomes painful. Or for those of us who have had shingles, we know that light touch in this area can be very painful. And the textbook view is that touch becomes painful due to a rewiring of the touch system at the level of the spinal cord. So the idea is that touch fibers gain access to the pain system at the level of the spinal cord. But we want to test the hypothesis that it's the ultra-fast nociceptors that become more sensitive following a peripheral inflammation. So we plan to do microneurography recordings from ultra-fast nociceptors and then induce a skin inflammation. And we will measure the skin inflammation with a technique developed by Rolf Sager at Wallenberg Center. And then we want to see if the ultra-fast nociceptors start to respond to light touch following the skin inflammation. They normally don't respond to light touch, but the hypothesis is that following a skin inflammation, they will start responding to light touch. That would mean that touch-evoked pain is signaled by sensitized nociceptors. So we don't need this more complicated working mechanism of the rewiring of touch. And if the ultrafast nociceptors become sensitized by inflammation, that might provide us with a new pharmacological target. And then the second project is the spinal cord projections. And pain is typically signaled in the spinal thalamic tract in the spinal cord, whereas touch is signaled in the dorsal column. So what we ask is, do the ultrafast nerve fibers project together with other pain fibers in the spinal thalamic tract, or do they project with touch fibers in the dorsal column? And to study this in humans, we have access to two patient categories. One is patients that will undergo anterolateral cordotomy. That is when you cut or lesion the spinal thalamic tract to help people with chronic pain due to terminal cancer disease, typically lung cancer. And the other group is patients that are going through, it's called midline cordotomy, where you cut the dorsal columns, and visceral pain is signaled in the dorsal column. So this is to treat very, very difficult, very, very severe visceral pain. And we have data suggesting that the ultrafast nociceptors are needed to discriminate between different levels of pain. So we want to see how well the patients can discriminate between different levels of pain before anterolateral cordotomy, as well as before midline cordotomy, and then compare it with their capacity after the lesions. So by doing this, we hope to be able to find out if it's the spinal thalamic tract or the dorsal columns that are necessary for signaling ultrafast pain and pain intensity discrimination. And we have already published a study on this, and it seems like it's the spinal thalamic tract that is crucial for ultrafast pain signaling. And when we know where in the spinal cord these ultrafast nociceptors project, we hope we can improve the methods of spinal cord, electrical spinal cord stimulation for pain relief. And we're also asking where in the brain the ultrafast nociceptors project. And here we want like to use fMRI, but it's quite complicated to just stimulate the ultrafast nociceptors without stimulating the slower A delta and C fibers. So what we plan to do is use the microneurography technique, but instead of recording from nerve cells, we would like to stimulate single nerve cells. So you can, normally we record from a single nerve cell, but we can also send current through the electrode and stimulate a single nerve cell. And this can be done in fMRI. It has been done by our collaborators in Liverpool. So it's technically possible, although difficult, of course. But the idea is to, in the MR camera, stimulate a single nerve cell and see where in the brain we get a bold response. And the question is if the ultrafast nociceptors project more to the somatosensory cortex, like touch fibers, or more to limbic cortex, like it's C fibers. And if we can find out the cortical projection targets for ultrafast nociceptors, we hope that we can improve the methods of repetitive transcranial magnetic stimulation for pain relief. And the last project is about finding a way to diagnose or measure from the pain system. And we are studying something called the nociceptive withdrawal reflex, and that is something you have all experienced as well, because if you step on something sharp, you will automatically withdraw the leg. And we try to mimic that in the lab by applying an electrical stimulation on the sole of the foot and record from the tibial anterior muscle. And this is a technique that has been used for 40 years, and it has been known already from the beginning that you get two responses, an immediate contraction of the tibial anterior muscle and a slower. And since this is a beta conduction velocity, and since the view is that it's that there are no pain fibers with this conduction velocity, this was thought to reflect a touch response, and this is the true pain response. But we would like to test the hypothesis that this is indeed a pain response, first early pain response signaled by ultrafast nociceptors. And this is the PhD project of Umi Torell. We also want to see if measurements of this nociceptive withdrawal reflex is clinically useful. So we are now studying patients with chemotherapy-induced neuropathy and patients with diabetic neuropathy. And we do a lot of tests in them when we ask for the subjective pain symptoms. Many of them suffer from chronic pain. We do a thorough neurophysiological and neurological examination, and we measure the nociceptive withdrawal reflex. And we ask if the threshold for withdrawal correlate with chronic neuropathic pain. If so, this could be a measurement from the pain system. And this is the PhD project of senior consultant in clinical neurophysiology, Salvador Amezcua. So what I've been talking about so far is the clinical part of a larger characterization of pain fibers. And the human studies are done by Saad Nagy, Umi Torell, and Salvador Amezcua. And then we have a large animal part, which we do in collaboration with Walker Jackson, Max Larsson, and Martin Stott here at LiU. And then it's Malin Lagerström in Uppsala and Patrik Elfors at KI. So with that, I think should we all questions at the end, or how do you?
I was thinking we could take questions at the end.
Yeah, so then I leave to Walker.
Okay, thanks, Håkan. I should also say thank you to Håkan and Max for inviting me into this project to begin with. And also thank you to Stefan and Rosalind for inviting us to come tell you about our project today. And thank you also to the audience for sticking around late in the day to hear what we're doing. Sorry, I'm slow to get this coordinated here. It's not working, right?
It was working. We could see your screen for a second. But you would probably want to share specifically the presentation. Yes. And yeah. Excellent.
Now? Okay.
Mm-hmm.
And now it's in presenter view or PowerPoint view. If something looks wrong, just let me know and I'll stop.
Looks great.
Okay, so my group normally studies neurodegenerative diseases, and the most famous neurodegenerative disease is Alzheimer's disease. But there's some things that are very interesting about these groups of diseases. And one thing in particular is this concept of selective vulnerability, where the diseases tend to target specific brain regions. And this is interesting because they're all caused by misfolding of specific proteins, and the proteins that are causing these diseases are not more abundant in the parts of the brain affected the most. And so why this happens is really a mystery, and some examples of why this is so unusual is certain diseases, and we have different diseases because they target different brain regions, and you can have two different diseases caused by different mutations to the same protein targeting different brain regions. And then on the other hand, you can have two different diseases caused by different proteins, but they had the same mutation. So in this case, it's a long stretch of glutamines. And so my group is really interested in why does this happen? And so how am I connected then with this project on pain? The pain project will involve a lot of experiments in animals, and one thing that my group brings to the table is some experience with engineering and working with transgenic mice. And transgenic mice are really useful for this because it allows you to control cells, and you can do it specific populations of cells. And one example is a method called optogenetics, where genetically engineered cells, so specific neurons, for example, in the brain or the spinal cord, could be controlled by pulsing light on them as long as they express the right kind of protein. My group also uses a different transgenic tool or set of tools called ribotag and tagger, and we use those for gene expression. So a brief outline of what I'll talk about today is an introduction to how transgenic animals are produced and some of the caveats with them. Also, a special method that's used with transgenesis, which involves recombinases. And then I'll briefly introduce these methods that my group is using. And the reason for introducing this is because we'll be using these methods in the project on pain. And then at the end, I'll give you an update about what we plan to do and what our progress is. So when making transgenic animals, there are two basic ways. One is called random integration transgenic, and the other is targeted. And so with the random integration transgenic, you, in this example, you would be making 3 different transgenic lines that have different versions of a common transgene. And the way this is done is these 3 different transgenes are introduced into an egg of a mouse, and then they integrate into random places in the genome. And this is marked by integration into different places and different chromosomes. The other way is you can make modifications to the same place of the genome and do it repeatedly, and you can know from previous experience that the expression between the different lines will be the same. And so now I'll give some examples of what happens when you go with the random integration approach. And in this image, this is an in situ hybridization of a mouse that's looking towards the left. So this is the front of the mouse and the top of the mouse's brain. Sorry, this is the front of the mouse's brain and the back of the mouse's brain. And we see, so these are 2 different founders of the same transgene. So the transgene integrated in different events and therefore different places in the genome. And we see that in founder line A, the transgenes expressed everywhere in the brain except for this region called the striatum. And then with founder B, it's expressed everywhere. Another example is using the same transgene vector as the previous example. In the top row are wild type mice. The bottom row are the transgenic mice. The first column is showing the expression of the transgene or the wild type version. The middle column is showing a cell marker for a special kind of cell called a Purkinje cell. And then the third column is a merge of these images. And the point with this figure is that this transgene is not functioning in Purkinje cells. However, we have transgenic mice using the same transgene vector that are very clearly expressing in Purkinje cells. And so what's happening is where these transgenes integrate in the genome influence where they're being expressed in the brain. One last example, and now it's a different transgene vector, but we have two transgenes that are essentially identical. The difference is one is making a green fluorescent protein and the other one has been mutated so that it makes blue light instead of green. And then these two mice are crossed, and then wherever both transgenes are being expressed, we see a light blue color. And when we look at a brain from such an animal, we see that some cells express only the green transgene, some cells express only the blue transgene, some cells express both transgenes, and a lot of cells express neither transgene. Now let's change this scenario just a little bit, and instead of expressing green fluorescent protein, let's have a disease model. And then instead of the blue protein, let's have a therapy. And then when we cross these two lines together, if the therapy did not rescue the disease, then we have to ask, did it not work because the therapy was a bad idea, or did it not work because it was not expressed in the right cells? So you can imagine if the green transgene represents the disease transgene and the blue one represents the therapy, you can't be surprised that therapy did not work because it's not expressed in the right cells. So to avoid these issues, we use a gene-targeted approach where we put transgenes into specific places in the genome. So a quick introduction into this tool called Cree-LOX system. And so when we are designing transgenes, we're often trying to make a reporter line. For example, those last mice I was just showing are expressing fluorescent proteins, which can be thought of as reporters. And in this configuration, this represents a transgene in the genome. There's a promoter that should be expressing or driving the expression of this reporter, but it can't because between the promoter and the reporter is this piece of DNA called a stop cassette. But there are sequences around the stop cassette called LOX sites. And then if we add an enzyme called Cree, it causes recombination to happen between these LOX sites, and that takes away that stop cassette, and now you can have expression. And so the magic with this for transgenic animals is you can control when or where, or both, Cree is being expressed. And so you can use Cree, for example, to be expressed specifically in these mysterious neurons that Håkan has discovered. And so that's a sort of an intro into what we're trying to go for with this project. Now there's a complementary system called flip recombinase, and it recognizes FRT sequences in the DNA. And they can be used for intersectional methods. And let's imagine we have a complex tissues. All tissues are very complex and they're made of lots of different kinds of cells. And they're represented here as the cells are represented in different shapes and different colors. And let's imagine that we've learned that in the round cells, they particularly express gene A. So gene A is expressed only in the round cells. And then we learned that gene B is expressed only in the blue cells. Well, if it turns out that we want to study specifically these round blue cells, we can do this by expressing Cree driven by the one gene and flip driven by the other gene, and breed them or express them in the context of a transgene or reporter transgene that is dependent on Cree and flip. And then that transgene will be expressed only in these cells that we want to study. So that's the first part of the intro. Now I'll get into the ribotag and tagger methods. And so the ribotag method is essentially a method to purify ribosomes, and it's a way to understand how specific cells are responding to a specific stimulus. And the magic is we can express it in specific cell types because it has these LOX P sites I introduced just a minute ago. So normally the transgene will stop at this exon, but if we add CRE and cells of interest, this exon goes away, and now the next exon 4 is going to be expressed, and this next one has an antibody epitope on it. And so if this CRE is being expressed only in the green cells, it'll make these ribosome proteins that have this antibody epitope on them. So now these bubbles represent ribosomes on mRNAs, and they have green dots because they're coming from the green cells. And then these other ribosomes, so this is looking at a homogenate, so a piece of tissue that's been ground to make a liquid. And so the uncolored cells over here, they have ribosomes without the green dots. So we purify specifically the ribosomes with the green dots with antibodies, and then we can sequence the mRNAs that are co-captured to understand how those cells are responding to these stimuli. And I just want to give an example of what we've done with this system before to demonstrate how sensitive this method is. And so we're studying a disease called prion disease that's initiated by injecting a material into mice, and it causes a terminal neurodegenerative disease 23 weeks after injection, and we're looking right now at data at 5 weeks after the injection. And each of these charts represents the electroencephalogram of mice at different vigilant states, so mice that are awake or in non-REM deep slow wave sleep or REM dream sleep. And then the groups are represented as red is the disease group and black is the control group. And at this early stage, at 5 weeks after the injection, the black and red lines completely overlap at all of these frequency spectra in all of these vigilant states. At 18 weeks, it's different, where we see an increase in the diseased animals specifically at these frequencies in all vigilant states. And this coincides with theta, and it becomes significant around the 18-week time point. And so we decided to say that the 18-week time point is our disease onset. And so it looks like it's a big difference, but I should highlight that these charts are broken to sort of emphasize the difference. So this is, in my mind, this is when a human might go to the hospital because something's not quite right. And the clinician might do an EEG and say, yes, there's something not quite right, but we can't be sure right now. And the person doing these EEGs, even at this 21-week time point, could not tell there was anything wrong with these mice. They still looked unremarkable. So we call this disease onset. Then to get a very early disease stage, we go back in time and see when the two groups are completely overlapping, and we estimate that to be around 10 weeks of age. So that was our early disease stage, is 10. So when we look at neuropathological changes at the 18-week time point, the mice look healthy. They have a subtle change by EEG. They do have neuropathological hallmarks of neurodegenerative disease, including aggregation of the disease-causing protein. Also, astrocytes and microglia begin to change their morphology. So clearly there are some changes going on here, even though the mice look normal. However, at the 10-week time point, you really can't see any difference. And this is our challenge. This is, in our view, this is when the disease and control groups begin to separate. And we want to use the ribotag method to understand what's happening at these very early disease stages. So we used ribotag to study 4 kinds of neurons as well as astrocytes, and we detected about 18,000 genes in all of our groups of animals, and it's a very high number. There's only 22,000 genes in the mouse genome, and we don't expect them all to be expressed in all cell types. And then when we look for the differentially expressed genes, we see at this disease onset time point, there are thousands of genes changing. And that was really a striking result because there is neuropathological changes and there is a subtle change by EEG, but the mice looked really fine. And to see that many changes was really surprising. And even more interesting is we saw changes at the very early time point where we can't see things any other way. Changes included circadian rhythm pathways and mitochondria and protein synthesis problems. So my group went on to make another mouse line that does a bit more. Now we can use it to capture ribosomes like the previous model. We can also use it to pulse label newly made RNA, label nuclei to study chromatin, or label the RNA-induced silencing complex to study mature microRNAs. And these are interesting because they regulate mRNAs either to target their destruction or to stall translation. And one last feature is this is one of only a handful of reporter mouse lines that was built in a way that can be used both Cree and FLIP dependently. So where are we with our project? So one of our contributions to this project were to make some knock-in mice expressing Cree at a NEFH locus, and then also some FLIP recombinase mice at a different locus. And these mice now exist. This mouse line currently exists at the ESL stage, but we expect to have full mice in the next few months. And then once we have these mice, we can validate them using the tagger system or ribotag. And then with that, we can identify differentially expressed genes that are induced by pain. And then this will give us a marker. And then we can use this marker to then characterize these ultrafast nociceptors that Håkan introduced to us. And I'm out of time, so I just want to quickly highlight some people that were involved in this pain project. And Lek is the one that designed and engineered the DNA and the constructs for making the new mouse lines. And he's done a lot of the work to characterize the new mouse line in the ES cells. And Xuan Yu has just recently joined the project, and he's going to continue on by helping Lek to phenotype these ES cells. And then eventually, once we get the mice into a position where we can use them, then he'll continue on to characterize them. And I'll stop with that. Oh, I should also point out, I have a fantastic group. I'm very grateful for them, and I'm grateful to be here in Liu and also for the Wallenberry. And thank you for your attention.
Thank you, Walker and Håkan. We'd like to open for great talks here. This very, very interesting topic. I'm sure that we have plenty of questions in the audience here, so I'm going to open up the floor. If you would like to ask a question, I think we'll just allow you to unmute yourself.